Hello everyone, welcome to another video of Biotech Academia. This is the part number 2 on the topic of what are genes. In the part 1 we have discussed some traits, some aspect, the introduction, function and structure of gene, and each and every aspect will be discussed in a detailed video by describing the each segment of that topic. And now in this part 2 we will also overview that what are the inheritance pattern according to Mendelian inheritance in a gene and genomic segments. We will also discuss that what are genes and evolution, and we will also look at the modern genomic technologies that are very common and that are used in advanced genetic research for the genetic evaluation. Now let's start with the genes and heredity inheritance pattern according to Mendel. So Mendelian inheritance named after Gregor Mendel, which describes that how traits are passed from parents to offspring through a dominant and recessive alleles. Now for those who don't know what are alleles, let me tell you that an allele is one of two or more versions of DNA sequence. It could be a single base or a segment of bases, and that are present on the genomic locations. Now, an individual inherits two allele, one from each parent, like for example, one allele from father and one allele from mother for any given genomic location which such variation exists. Now, what Mendel said about the inheritance pattern? Mendel said that, of course, he did experiment with the pea plant, and what he said that the sex-linked inheritance involved genes located on sex chromosome X and Y. Now, as we know that a woman has XX pair of chromosome and a male has XY pair of chromosome. For example, color blindness is more common in male because the gene responsible is located on the X chromosome. So male have only one X chromosome, so the single recessive allele will result in this condition. Now, what else Mendel did about this? In the diagram, you can see that Mendel said we can predict the possibility and the probability of offspring inheriting certain traits. Like he said, we can predict which traits are inherited from father and which traits are inherited from mother. For example, if both parents have heterozygous allele, their offspring have 75% chances of showing the dominant trait and a 25% chance of showing a recessive trait. You can see this in the diagram, the father carrier and the mother carrier. So what Mendel did, Mendel give us the statement that we are very probable and we can predict the possibility of offspring that are transferred from parents to offspring. Let's see what are genes and evolution after Mendelian inheritance. You may be clear about that. What is the probability of transferring gene from parents, from for example from father and from the mother. Let's look at the genes drive evolution. So evolution is a process by which the population of organisms change over time through variation in their genetic makeup. Here we can see that evolution can arise from natural selection. So the natural selection is the key mechanism of evolution. It acts on genetic variation within a population, and it also favors the traits that enhance survival and reproduction process. Let's take an example of a papered moth during the Industrial Revolution. Now dark color moths were better camouflage against root covered trees, so they were more likely to survive and produce, increasing their frequency in the population. Other mechanism includes genetic drift and gene flow, and that genetic drift is a random change in allele frequencies in a population, more significant and sequent in a small population. For example, a natural disaster might randomly eliminate the individual with certain traits. Speciation is a process by which new species arise, and it occurs when population of the same species become isolated and diverge genetically and over time. Now, factors like geographic isolation or different selective pressures led to the emergence of distinct species. Here, Charles Darwin, the islands of classic example tell us that different islands have different environments, and that environments lead to the evolution of diverse peak shapes adapted to their specific food sources. So we can say that understanding the gene and heredity pattern helps us predict how traits are inherited, while evolutionary mechanism explain how species change over time. What we said, that genes and heredity patterns, it help us to predict how traits are inherited from both parents, and the evolutionary mechanism explains how these species changes over time, for example from the very beginning to the very end. These concepts are fundamental in the field of genetics. Extension tells us about the difference of that species and difference of that organism from that evolution to the current condition. Now let's dive into the modern genetic technologies. And these modern genetic technologies are the innovation in sciences, and these innovations have transformed biology and medicine, which offers the insights and capabilities of the subject. Today we will explore the DNA sequencing, including Sanger sequencing, next generation sequencing, with the just overview, because a detailed lecture is coming very soon, as well as CRISPR and gene editing technology, how gene editing technology and CRISPR, for example modern technologies like DNA, Sanger sequencing and next generation sequencing has revolutionized the genes, how it impacts the human, and how their impacts would be for future. This will be explained in the upcoming detailed video. Today is just the overview, and I'm telling you in this that how the things work, how genes are evolved, what are different traits, what are different segments, what are the technologies that involve these days. So let's begin with the DNA sequencing. So, which is the process of determining the exact sequence of nucleotide within a DNA molecule. You know that DNA is made up of nucleotide. So, what is DNA sequencing? It is a process of determining the exact sequence, like the really exact sequence of nucleotide within the DNA molecule. There are two most common technologies that come in DNA sequencing. To sequence the DNA, two more technologies are common, that is Sanger sequencing and next generation sequencing.